morning, guys. I am Dr. D.R.T.SUMATHI. Right now I am going to take the developmental biology. Next. This subject also, first semester zoology, subject developmental biology, the unit also second unit. Already I have issued first module in first unit, and this module is a second module for second unit. Next. This unit we have to know about fertilization. You know the fertilization very well about all the students. That is one of the interesting topic also. First we have to know about what is sperm, what is egg. Another one is that is the formation of zygote. The formation of zygote, after that producing the young ones, that in between time also called fertilization. Just we have known about where the fertilizations occur, what is the organs important for fertilization. That is important to knowing about the fertilization and developing of the embryo within our body. This is the fallopian tube is one of the important organs to formation of fertilization. This picture shows fallopian tube, ovary and fimbriae, uterus, cervix and vagina. What is the role of fallopian tube? We have pair of fallopian tube and a pair of ovary also. This is our body also very amazing part in our body. That is because the ovary also sponge shape. Each and every month also the egg is formation in the one part of ovary. Another part of ovary next month formation of egg. What is the role of fallopian tube? After formation of the egg also going to the fallopian tube. When the egg is going, the egg and the sperm also induce, sperm also fuse with egg, the formation of zygote. The zygote is moved to the fallopian tube. Which one is here help? What is the role of fimbriae? That fimbriae also just capturing the zygote. After that it goes to the fallopian tube. After the fallopian tube, 2 to 3 days, 4 days, the egg also developed in the fallopian tube. The cell stages, next cleavage stages also occurs in the fallopian tube. It goes to the uterus. The role of fallopian tube also passes the zygote to uterus. The fimbriae also is very important. Next one is the uterus, another one is the cervix, that is the vagina. Next. Already I told you about the fimbriae. What is the role of fimbriae in this part of the fimbriae? External structure is a finger-like projection on the end of the fallopian tubes and the oldest ovaries. That is a matured ovaries. That's all. Each month the fimbriae catch the egg of your ovaries and release during ovulations. In case the sperm is not fused to the egg, then normally the ovulations occurs. Normally menstrual cycles also occur. And our fimbriae sweep the egg. Sweep the egg means ovum into the fallopian tube where the egg can be fertilized. The fimbriae are just one small portion of our fallopian tube, but they play the significant roles in the conceptions. That is, conceptions is used for family planning. It is used of the conceptions. And the fimbriae and the ovary also joining the fimbriae, and also it goes to the fallopian tube of the egg. The role of fimbriae. Next, this is an amazing picture. Just we have to know about the what are the stage occurs in the during fertilization. The first, the ovary, that is the ovary also. The picture shows the ovary. Next, the ovulations occurs, and the fimbriae finger-like projection is called fimbriae. The egg goes from ovary to fimbriae. Already I told you the fimbriae role is just capturing the egg. After it goes to the fertilization, and during fertilization, the egg, the sperm also fuse and formation of the zygote. Next stage also, zona pellucida stage, that is your first cleavage stage. The egg after zygote, formation of cleavage. That is the first cleavage, and also occurs in the oviduct. Next stage also 2 cell stage. 1 cell also developed into 2 cell, next 4 cell, next 8 cell, next 16 cell, next 132 cell after the formation of morula. Morula, blastula, gastrula, that is all the stages also developed. Everything also occurs in the fallopian tube. That's very important. After the blastocyst and early stage of implantation occurs in the uterus. That is the amazing of fertilization. This picture shows development of human embryo from fertilization to implantation. Implantation means implanting of the after developing of these cells goes to uterus. That is the implantation. Next, normally fertilizations can be defined as we have known about very important word. What is meant by fertilization? Union of two haploid gametes. That's very important. Why we call it haploid? Egg also haploid and sperm also haploid. One N, normally N. The N is also formed 2N, that is a diploid stage. The spermatozoa and oocyte. Spermatozoa means sperm, oocyte means egg. Here to refer to the egg to restore the diploid state from a zygote through the process of egg activation. Fertilization can be defined as the union of two haploid gametes. And that haploid gamete also spermatozoa and oocyte. That hence these mitotic divisions, the result the cell differentiation and embryo also development. After formation of zygote, the embryo development. During fertilization, the sperm and egg unit one of the fallopian tube and form a zygote. These fertilization also occurs in the fallopian tube only. The fallopian tube also very important. In case sometimes the egg also developing stages also develop some disturbance occurs in the fallopian tube. Just we have to removing the zygote. Next, then the zygote travels down the fallopian tube where it becomes a morula stages. Already I told you 2 cell stage, 4 cell stage, next to 13 cell stage, another one is a blastula, another one is a gastrula, another one is a morula stages. Till morula stages, the egg also developed in the fallopian tube. Once it reaches the uterus, the morula became a blastocyst. After that, the egg also goes to the through the fallopian tube and it goes to uterus. That uterus also the cells developed. The cells also called blastocyst. After that, the formation of another organizations, organs also developed. That is called organizer. Organs also developed within the uterus. That's very important. The blastocyst then burrows into the uterine lining. A process is called implantation. Already I told you the implantation. The blastocyst then burrows into the uterine wall. That uterine lining, that is the uterus wall. The process is called implantation. What is the stages can be the formation during fertilization? We have to divide at 4 stages minimum. So many stages occur. Mainly we have to divide at 4 stages. First one is a sperm preparation. Another one is a sperm egg recognition and binding. That stage also formation of zygote. Another one is here the sperm egg fusions. Next stage is here fusion of sperm and egg pronuclei and the activation of the zygote. So what are the role? What is important for fertilization? Sperm preparation and also egg also coming that places. I mean it is coming from that ovary to fallopian tube and the sperm and egg also fusions. That very important. The fusion of the sperm and egg pronuclei and activation of the zygote, because nuclei also very important. Only haploid nucleus also occur. The haploid nucleus is formation of diploid. That is formation of zygote. That's very important of the yolk material. Next, there are 2 different types of fertilization occurs in generally. Not only human being, in generally 2 types of fertilizations occur. One is the internal fertilization, another one is the external fertilization. You know very well about ourselves, our human being also only internal fertilizations. What is the process occurs in the internal fertilization, where the fusion of egg and sperm occurs inside the female reproductive tract. That is called internal fertilizations. During that example also, example also human being. What is meant by external fertilization? Just opposite, where the fusion of the male and the female gametes occurs outside of the body of the organization. For example, sea urchin, frog, etc. But what are the external fertilization? How it occurs? The egg also released, sperm also released in the water. After that, externally, within the water, the egg and sperm also fused and formation of new ones. That is a very good example also. Frogs. Now you know the frog also amphibians. Next, just we have known about what is the fertilization scheme. So some of the words also very important, because we have to know about the definition of the words. We have to know about the full meaning of the fertilization. Otherwise, it's very difficult. What is in the way? Blastula, blastomia, gastrula. That's also very difficult. Just you have to know about the important words. That words also called key points. And the first one is here blastula. Another one is here blastomia, miroblastic, holoblastic, inner cell moss, gastrulation, trophoblast, blastulation. Everything also. What is meant by blastula? 6 to 32 celled hollow structure to formed after the zygote undergoes a cell division. Already I told you 2 cell stage, 4 cell stage, 8, 16, 32. That is called 6 to 32 cell hollow structure. That is called the blastula stage. Another one is a blastomia. After formation of zygote, the cell division occurs. We do not call about any one of the cells development cell division. That stage also called, cells also called blastomia. Any cell that result from division of fertilized egg, that cells also called blastomia. Another one is a miroblastic, undergoing only partial cleavage, that is called miroblastic, not fully cleavage. Cleavage also, so many patterns also. Next slide we have to know about the what are the type of cleavage is known. Just you have to know about miroblastic. Another one is the holoblastic, and the holoblastic means cleaving and separating into separate blastomeres. The formation of separate blastomere, the opposite of miroblastic. Another one is the inner cell moss. But during cell moss, the moss of the cell within a primordial embryo that will eventually developing. Formation of eventually developing, that is formation of very distinct character from the fetus, the most of the eutherian mammals, that is a placentation mammals. The inner cell moss also occurs, that is a eutherian mammals. Another stage also gastrulation. During gastrulation, the stage of the embryo is development at which the gastrula is formed and blastula by inward the migration of the cells. Gastrula, next stage also blastula stages. Invagination, inward of the migration of the cells, the movement of the cells, that's all. Trophoblast, that the membrane of the cells that form a blastocyst during the early pregnancy to providing nutrients of the embryo and developing the part of the placenta. The formation of placenta also very, very important, because our nutrients also goes through the placenta. That the trophoblast cells also occurs give the nutrients from the embryo. Next, these are the cleavage. That's very interesting and very important. We have known about after the fertilization. Already we have known about the key point also, holoblastic. That the holoblastic also have the isoleucithal. Another one is here mesoleucithal. Already we have to know about the macroleucithal, microleucithal, mesoleucithal, isoleucithal, and the centroleucithal, everything also. How we differentiate from this leucithal mean content of the yolk present in the animal pole and vegetal pole, that is called leucithal. Animal poles also contain some of the egg content, vegetal poles also contain egg yolk material. Normally a very good example of the insect, that is the content the centroleucithal. Leucithal means the content of the amount of yolk material present in the egg. This also holoplastic nature, that is the isoleucithal. Radial, normally radial cleavage, for example echinodome and amphibians also. The cells also divided into 2 and 4 cells and also 8 cells. Another one is spiral shape. The picture also shows the cleavage also spiral shape cleavage. For example, annelids, mollusk and flatworms. That the annelids and mollusk also have this type of cleavage. 2 cell stage. After the cleavage is occurs in the 4 cell, that is the spiral shaped. Next stage also bilateral. Bilateral symmetry, another one is a bilateral cleavage, that example tunicates. That type of cells, animals also have this type of cleavage. Bilateral, 2 cell and 8 cell stage and 16 cell also, that is a bilateral. Another one is a rotational, that is very important. The rotational cell divisions, example our body, mammals and nematodes also. 2 cells and 4 cells, 8 cells, the cells also rotational cleavage. Another one is a mesolicital. In radial amphibians also, the mesolicital cleavage also occurs. It's a very good example. Next. Normally, in holoplastic cleavage also, otherwise called total cleavage. What are the functions occurs in this cleavage? Mean when the cleavage furrows divide the entire egg also divided. It may be equal, another one is unequal. That entire cleavage also occurs equal number of the cleavage of the cell division, another one is unequal. In equal cleavage, when the cleavage furrow cuts the egg into 2 equal cells, 2 equal halves, it may be radially, it may be radial symmetrical, bilateral symmetrical, another one is a normal symmetrical, another one is a spirally symmetrical. Maybe it is a irregular in shape. It may be chances these equal shades. That unequal means when the result, the blastomere becomes unequal in the all the cell division, not radially, symmetrically, bilaterally, no one occurs. All the cells also dividing unequal size, not equal distribution of the yolk material, equal distribution of the cells also, everything also unequal. The holoplastic cleavage is otherwise called total cleavage. These cleavage also divided into 2. One is a equal cleavage, another one is a unequal cleavage. During equal cleavage, already we show about the picture, symmetrical, bilateral, radial and spirally symmetrical, everything, any one of the shape also occur in the equal cleavage. But unequal cleavage, the above structure is not occurs. The above cleavage is not occur. Next. This is a meroplastic, the continuations. In pilolicital, the bilateral cephalopod mollusca also, and the cleavage also occurs. Another one is a discoidal, and the discoidal means example fish, reptile and birds. The discoidal shape also occur. Centrolicital, the superficial, and most of the insect, the centrolicital. Already I told you, the centrolicital occurs in the insect. That's very important questions for TRB also. What are the lecitals occurs in the insect, that is called centrolicital. That's very important. Next. In meroplastic cleavage, when the segmentation takes place only in the small portion of the egg occurs and it is a formation of the blastoderm. It is called meroplastic cleavage. The cells also called blastoderm. During cleavage, after that we called blastoderm. Usually, the blastoderm is present in the animal pole, another one is the vegetal pole, and the yolk also remain uncleaved state. Normally, the plan of division does not reach the blastoderm and the blastodisc stages. That's very important. On this cleavage, normal occurs in the animal pole, another one is the vegetal pole. Upper region is called animal pole, lower region of the egg is called vegetal pole. Next. And that is a transitional cleavage. In many eggs, the cleavage is there atypical, which is neither typically holoplastic nor meroplastic. It may be assumed the transitional stage between the holoplastic cleavage and another one is a meroplastic cleavage. It is maybe transitional cleavage occurs in the most of the eggs. And the most of the animals, normal this development also, cleavage development is also very important. After fertilization, the cleavage also recognize, that is important for formation of organ in our body. Most of the animal proceed through these stages during the development. First stage is the zygote, after the formation of cleavage. That is a cleavage is a 8 cell stage. Another one is a blastula. The picture shows a blastula. The formation of blastula and the formation of seal, C-O-E-L. Seal means cavity. That means that's the blastocele. Another stage also called gastrulation. Yes, you have to differentiate this diagram. Blastomere and the formation of blastocele. The cells also invaginate inside of the invagination during the gastrulation. After the blastoceles and gastrula, formation of arcandiron, endoderm cells, ectoderm cell and plastophore also formation. Endoderm cells is the outer layer of the cells, ectoderm is the inner layer of the cell. Upon these early cleavage stages, blastula and also gastrulation. During gastrulation, it established a germ layer. That is, a germ layer means endoderm, ectoderm cell. That's all. Body plan, the segmentation. So, what is the segmentation body occurs in invertebrates? It involves a neurulation, formation of nerves. That is called neurulation. Another one is a morphogenesis. That is, a morphogenesis means formation of organs. Organogenesis is called morphogenesis. Which organ is formed? First, eye development, brain development, next organ, internal and external organs also developed. After cleavage, the organ also developed in our body. That's why the cleavage also very important. It is a very good picture. The blastula and gastrula, we have to differentiate. Gastrula also the blue color outer cells also called ectoderm cells. Another inner green color cells also called endoderm cell. Within the violet color cells, that places is called acantidon. And this base of the cells is called plastophore. The formation of cavity, that is called blastocele. In the blastula and also gastrula. After developing of the blastula, that stage also called gastrula. That is already I told you, amazing picture in the invagination. The cell invaginate, the yellow color picture also, the cells also going to inside of the cell. That is called invagination. Another one is a involution. The cells also differentiate from this particular just invaginate. That is a involution. Another one is a cells goes to inside. That is called incrustations. And the delamination. Another one is a epipoly. The epipoly means cell is the outer side, and that is two type of poly. One is a M-poly, another one is epipoly. In epipoly, the cells also invaginate in the outer side. A single cell zygote will undergo a multiple round of cleavage or any one of the cell division. It produces a ball of the cells. That is, a ball of the cell means there is no organ developed. Only cells also form it in the ball shaped cell. That stage is called the blastula. And that during that blastula stage, it filled with some fluid. That is a cavity. Already I told you that is called blastocele. The cavity is called blastocele. Any one of the cavity occurs in the body, not only cleavage, the cavity also contains a fluid that is called blastula, that is fluid, blastocele fluid. The blastula eventually organizes itself to two layers, that is one inner layers, another one is the outer layer. The innermost layers is become a embryo, outer trophoblast outer layer became a placenta. The structure is now, it is called that the inner layer and outer layer blastula structure is called blastocyst, because the cyst also formation, so that is called blastocyst. During gastrulation, the blastula folded itself to form a three germ layers, that is called ectoderm, endoderm and mesoderm. Normally it will give rise to an internal structure of organization. The organization is also one of the important part, formation of organ in our body. The base also, which one is used to formation of organ in our body. These type of three germ layer. The germ layer also one of the very important questions, that is the formation of germ layer, ectoderm, endoderm and mesoderms. Next. Normally gastrulation in frog embryos is initiated on the feature dorsal side. The embryo just below the equator region, and that region is also called grey crescent region. That grey crescent is depends upon yolk material present in the places. You know the crescent shape in the moon, that is called grey crescent. Here the cell also invaginate to form a slit like plastophore. Plastophore, plastoderm, everything also related to the embryo formation. The cells also changes the shape dramatically. Totally they have to changes the shape during after the gastrulation. What is gastrulation? In the process of gastrulation, the frog epipoly, M-poly and invagination will take place. And then that all, what is in the epipoly, M-poly and the invagination? Everything also depends upon the cells also going to inside and outside. During gastrulation, the germ layer around the embryo, next step organogenesis is formed and created the sperm, egg and embryonic cell. As the days, the hours of the progress, it is also occurs, the zygote is starting the multiplying and also eventually many cells and rearranging the cavity. That cell is called blastula stage. Next. How the gastrula is formed? The gastrula is developed from the hollow single layered ball of the cells. That is called the blastula, after which itself the product of the repeated cell division or cleavage of the fertilized egg, the gastrula is formed. This is a very important to developing from blastula to gastrula. That is one of the scientists also, Lewis Wolfer. It is not birth, marriage or death, but gastrulation, which is truly the most important time in your life. Why? Because the gastrulation also formation of ectoderm, endoderm and mesoderm cells. After that formation of these type of cells, we getting the different type of organ in our body. So the scientists say like that, everything also important in our body, birth also, marriage also and death also, but the gastrulation also very important in our life. It's also recognized, it also decided the cell movement, the massive reorganization of the embryo and the formation of the simple spherical cells in the blastula and the formation of the multi-layered organism in our body. During gastrulation, many of the cells are near the surface of the embryo move to a new, more interior location. The primary germ layer also formed during the organizations of the gastrulation also formed. Different role of the ectoderm and endoderm, mesoderm cells also. Already I told you the role of cells, ectoderm, endoderm. Normally, endoderm cells is a germ layer form a gut region and internal organ also. Ectoderm cells, the role is most exterior germ layer form a skin, brain and nervous system and other external tissue. What is the role of mesoderm means? The middle germ layer, that is a mesoderm, it form a muscle, skeletal system and circulatory system also. Why? Because we call the gastrulation is important because all the organs is amazing to formation of our body after during gastrulation. So the gastrulation is very important in our body. Next. Then normally in this slide shows gastrulation of the frog resemble gastrulation of the many other animal, and I told you that 3 layers also formed and the difference of the frog also. One of the example, the human being also developed the large amount of yolk material in our body. The cells also end up to formation of yolk plug stage and the blastocele stage also formation, but does not make much difference time of the gastrulation process. Next, the process also occurs, our organs also develop, so gastrulation occurs in all the animals also. Next. Normal gastrulation, early embryonic development, most of the animal, it recognizes the trilaminar structure of the gastrula. The epipoly consists of the epipolic movement and empoly also occurs in the empoly movement. The embryo is here converted the double-walled cup-shaped structure. After that, the formation of new internal wall lining, that is called arcandina. The newly formed cavity also developed in the gut regions. After that, formation of another stage of the body, another organs also developed in our body. The cells also occur, the epithelial cells also spreading and multiple ways to formation of cells within the body. The empoly, simply movement of the 3 germ layer. Next one is the arcandina, the exterior of the blastophore and the future anus of the deuterostome. Instead the cells, the animal pole move down the embryo surface and the region is derived the gray crescent, the move of the interior region. In these stages, the morphogenetic movement is called epipoly and empoly. The movement of the cells during gastrulation in inner side and outer side, that morphogenetic movement is also called epipoly and the empoly stages. During gastrulation, the inward movement of the invagination is accompanied the cells. The change of the shape first became flask or bottle shaped. So most of the moss actually below the surface of the cell Already I told you mass of the cells is formed, ball-like structures is formed. Next stage is also formation of sinking into the interior, and they lose the remaining connection the other cell surfaces. That is the next stage is formation of shape, and also it cells, organs, everything also changes during gastrulation, and the movement of the cells also invagination. That's very important. That is a gastrulation of the stages. The picture show invagination. A sheet of cells called the epithelial sheet bend inwards. That's the basal side and the apical side. Incrustations. Individual of the cells leave an epithelial sheet and become freely migrating mesenchymal cells. The cells also goes to that mesenchymal cell. Another one is a involution. The epithelial sheet rolls inward to form a underlying layers. This is a very nice picture. These pink color cells also bending, and blue color cells also organizing formation of bend, formation of bending. That is called involution. That is already told you. Avipoly in the sheet of cells spread by thinking. Intercalations, that is a movement between another creating array of the cells. Convergent extension, rows of cells intercalate, and interaction of the highly directional. That is the avipoly, intercalation, and the convergent extension of the cells. That is a cell formation of cell. That is a very important of the fat map. Why we know about the fat map? The fat map diagram, the blastula shows the cells, the fat to the become a ectodome. Normally, just we have to differentiate from this color of the fat map. In the blue color and green color cells, red color and yellow color cells also. Normal, just you have to imagine the shape of the egg. Upper layer also called animal pole, lower layer also called vegetal pole. The blue color also shows blue and green color cells, fat is to become a mesodome. And red color cells also, the fat map to become a endodome, and ectodome cells also blue color, green color cells also mesodome, and red color cells also mesodome, yellow color cells also endodome cells. This picture shows neural plate also, nervous system also formation in the green color. And what is the role of epidermis? Already I told you, formation of skin. Mesodome, what is the role of mesodome? Muscles, kidney, bone, and gonads is formation. Endodome cells also formation of digestive organ in our body, and development of the lungs also. So just we have to know about what is the role of egg content within the cell. That is called fat map. Next, fat map is a diagrammatic representation of the prospective fat of each part of an embryo and the early stage of development. Why? Because the fat map diagram, the egg and the blastula indicating the fat of each cell and the region, and the later the development of the cell. Why the fat map is important? It is the essential tool in the embryological experiment. Why it is important for embryological experiment? These particular cells is used for this particular formation of ectodome, endodome, mesodome, and the organ development. So the fat map also very important. They provide the researchers and information. The portion of the embryo will normally become which larval and the adult structure. This embryo development also decided to formation of new organ, development of new ones, adult in our body. Next. Thank you.